Hi everyone, I'm Karen, and my project's topic is on the neuroendoimmune axis and how it contributes to neurodegeneration. When we learn about physiology, we often think about how each organ system works as a discrete unit. However, it has been shown that there is actually a lot of bidirectional communication going on between these organ systems through cellular and molecular mechanisms. The neuroendoimmune axis is made up of the nervous, endocrine, and immune systems. By using bidirectional communication through shared mediators such as cytokines, hormones, neurotransmitters, and neuropeptides, this group of systems is able to work together to maintain homeostasis in our internal environment in the presence of daily physical and cognitive stressors and even anticipated stress. The stress response is initiated by the hypothalamic pituitary adrenal axis, or HPA axis for short, which ultimately releases glucocorticoids to trigger acute stress mechanisms such as anti-inflammation and immunosuppression. Then why is it that we see chronic inflammation and infiltration by immune cells as a key characteristic in neurodegenerative diseases, even with elevated glucocorticoid levels? With chronic elevated glucocorticoids, tissues and cells begin to downregulate their GC receptors, reducing their sensitivity and leading to what is known as glucocorticoid resistance. Chronic stress has been shown to prime key areas of the brain for further neuronal damage. A study done by De Pablos et al. Showed that areas of the brain that are not as susceptible to inflammation, such as the prefrontal cortex, can become more susceptible to inflammation in the presence of chronic stress in rat models. In this study, chronic stress was combined with lipopolysaccharide, or LPS, and they were able to enhance microglial activation more when combined with chronic stress compared to controls. And there was a reduction in astrocyte numbers near the LPS injection site as well. Another study by Dufour and McBride showed that normalizing glucocorticoid levels in transgenic mice can partially revert symptoms of Huntington's disease, such as muscle wasting, weight loss, and reduced neurogenesis. The experiment also saw reductions in mutant huntingtin, or MHTT, inclusion bodies in key areas like the striatum, cortex, and hippocampus, thereby reducing molecular pathologies of Huntington's disease, such as autophagy inhibition and ER dysfunction. Many neurodegenerative diseases have mechanisms involving cytokines and immune cells, mainly for microglial activation, but other glial cells too. Cytokines are seen as a double-edged sword. On one end, they are vital in acute inflammatory responses and allow communication between immune and non-immune cells. However, when they are dysregulated, cytokines can recruit mediators that could further damage the body and are the key component of neuroinflammation. When microglia are activated, they release pro-inflammatory cytokines such as interleukin-1, interleukin-6, and TNF-alpha, along with anti-inflammatory cytokines, reactive oxygen species, and cytotoxic factors. The pro-inflammatory mediators can go on to modulate the blood-brain barrier, recruit leukocytes, and induce astrogliosis. Interleukin-1 beta and TNF-alpha, for example, can go on to cause glutamate excitotoxicity, damaging neurons and glial cells. Neurons damaged by microglia release factors such as adhesion molecules and trophic factors, which may amplify neuroinflammation. How can researchers take advantage of the connection between these systems? Since there are pathways that promote neurodegeneration, there must be pathways in which neuroprotection can be achieved. For example, growth hormone is secreted from the anterior pituitary and is involved in brain development processes such as neurogenesis, myelination, and synaptogenesis, but it also regulates IGF-1. IGF-1 also promotes systemic growth in the bone, muscle, and neurogenesis. A study done by Ramsey et al. Showed that chronic injection of growth hormone improves spatial learning and cognitive function in aged rats and rescued their plasma IGF-1 levels. Another neuroprotective pathway is the GABA and calcium mechanisms. GABA is responsible for regulating calcium influx in neurons, therefore they stabilize neuronal firing. There are still conflicting studies on whether GABA is neuroprotective or not. One study showed that elevated GABA from reactive astrocytes in the hippocampus may contribute to cognitive impairment in Alzheimer's patients. However, another paper showed that low doses of mucimol and diazepam, which are GABA receptor agonists, prevented spatial learning and cognitive impairments in Alzheimer's disease rat models. Modulation of the neuroendoimmune axis is still a growing discipline, and it is difficult to summarize in just one poster how vastly it affects neurodegeneration and repair. It'll be interesting to see how new findings will change how researchers see treatment options for prevention or progression of neurodegenerative diseases. I hope that through my presentation I was able to convince you of the communication that occurs through these systems. Thank you.